I'm Thomas Siegel-Shapiro.
And we're from Rice University, a small liberal arts school in Houston, Texas. Our project is quorum-taxis, programming E. coli to eavesdrop, stalk, and kill B. subtilis. This is our first year participating in the iGEM competition. So over the summer, we spent some time reading journal articles to see what kind of work has been done in the field of engineering bacteria so far. And one journal article that stood out for us in particular and inspired our work is one from UCSF, in which E. coli were engineered to invade cancer cells. And so we wanted to ask the question on whether or not we would be able to generalize and broaden this idea so that E. coli could seek and destroy another species or strain of bacteria. And so our work, quorumtaxis, integrates two pre-existing bacterial functions, quorum-sensing and chemotaxis, both of which are two-component signal transduction systems. Quorum-sensing is a method by which E. coli, or bacteria, is able to sense its own population density via signaling peptides, pheromones, that are released into their environment. And chemotaxis is the directed movement of bacteria towards or away a chemical in their environment. And so integrating this, we would have quorumtaxis, in which one strain of bacteria, in our case E. coli, is able to detect the pheromone released by another, in our case B. subtilis, swim towards it, send out a kill signal, and eliminate the second strain of bacteria. And so in designing our quorumtaxis system, one question we wanted to ask was, what is a good pheromone to use for our system of quorumtaxis? Well, the one system that has been well studied is the one in gram-negative bacteria, which is acyl-homoserine lactone, AHL-based. These pheromones are metabolic products and therefore have limited diversity. On the other hand, pheromones released by gram-positive bacteria, such as B. subtilis, are oligopeptides that are genetically encoded for and therefore have a tremendous potential for diversity. And so this gram-positive system that we're going to use from B. subtilis is a peptide-based quorum-sensing system that has 4 protein components. The first of which, COMX, is a precursor protein that is in the cytoplasm. It will be modified by COMQ via cleavage and farnesylation. The mature COMX moves out to the periplasm, where it's able to bind to COMP, a membrane protein. This in turn activates COM-A and therefore leads to transcription of genes, which allows the B. subtilis to sense its population density. And so, like I mentioned earlier, there's a large diversity in the COMX system. And if you look at the sequence alignment, and here we have the sequence alignment of the C-terminus of COMX precursor, this is just within various strains of B. subtilis alone. So you can imagine the diversity in other species of bacteria. There's little sequence alignment within this C-terminus region, and you can see mainly only the tryptophan is conserved. And so this diversity allows for orthogonal COMX systems to evolve that eliminate crosstalk and therefore reduce communication and background within species of bacteria. And so the COMP is the membrane protein that binds the COMX that is produced. And the COMP receptor is related to methyl-accepting chemotaxis proteins, MCPs. They're both related in that they involve a peptide binding to a transmembrane protein, and then this results in signal transduction pathways. And so E. coli has a number of these MCPs, and you can see from the table that depending on which protein is made, results in taxis of E. coli to a variety of chemicals in its environment. And 2 proteins that we're working with, COMP isolated from B. subtilis and TSR isolated from E. coli, have around 11% identity, which I'll talk about this significance in a couple of slides. So the TSR, one of the MCPs in E. coli, has a modular architecture, which allows us to play around with the system and recombine it pretty easily. And so it has a signaling domain in the cytoplasm that is pretty well conserved throughout, and a ligand binding domain, which is pretty divergent. And the variability of this region allows for the binding of various peptides. And that's what contributes a lot to the diversity. And so like I mentioned earlier, the COMP and TSR proteins have around 11% identity. So you may ask the question, well, if it's so divergent, how are we able to combine these into a functional receptor? Well, previous studies have shown that MCP-like proteins have been recombined to create novel functional receptors. And the divergence ranges from 3% identity of TAR and human insulin receptor to 58% identity between TAR and TSR. And so our COMP and TSR sequence identity of around 11% has a good possibility of creating a functional chimeric receptor.
Okay, so we've talked about our background information, kind of why we're taking this approach and why we think it has a good chance of working. Now we're going to start going over the strategy, or how we actually plan to implement this process and create our quorumtaxis system. Phase 1 is kind of the seek phase. It's where we work on making our bacteria seek out the B subtilis strain. And phase 2 will be the destroy phase, where we engineer the kill response. In phase 1, there are two separate parts. The first is using biobricks and our chimeric protein to actually assemble a working quorumtaxis circuit inside E. coli. And then the second part is building a chemotaxis model and adapting it for our new chimeric protein and using that to characterize and optimize our output. And then once we get onto the destroy response, we just need to extend our circuit so that we get a kill output once it's close to the target bacteria. So the way that we plan on creating our quorumtaxis is through a chimeric receptor. And this has a couple of advantages. First, it will let us keep the cytoplasmic domain of the chemotaxis protein from E. coli, which should let us preserve the adaptivity and sensitivity of the already existing chemotaxis system. Second, as we said earlier, there have been lots of examples of chimeras like this working out, so we think that we'll have a very good chance of creating a functional one. However, while we hope that our functional chimeras can exactly copy the cytoplasmic domain of the chemotaxis protein from E. coli, it's likely that it'll introduce subtle differences into how the protein works, which will then change the chemotaxis response of our E. coli. There are a couple of different ways in which our protein could differ. It could weakly bind the chemotactic attractor, in this case, COMX. It could bind the attractor but not transmit the signal as strongly. Or it could bind the attractor and transmit the signal but then not adapt as the normal chemotaxis system does. And in order to kind of examine these different possibilities and see how that will affect our output, we're modeling chemotaxis and looking at it with a mathematical model. To model chemotaxis, we first need to look at exactly what the phenotype is. Just briefly, E. coli normally move through a series of runs and tumbles. Runs are straight motion, and then tumbles will disorient it and move it in another random direction. If there's no attractor, the average length is about 1 second for each run. If the E. coli is placed in a chemoattractor gradient, the chemotaxis system lengthens all the runs that move it up the gradient and shorten all the runs that move it away from the gradient. This has the net effect of moving the bacteria up the gradient. The really amazing thing about the system is that it's very adaptive, meaning that if the bacteria is in a concentration of chemotactic attractor with no gradient, it will move with its average length of 1 second. So if the bacteria is in a plate with no attractor, it'll move with 1 second. And if it's in a plate with a high concentration of attractor but no gradient, it will still move with that same run pattern. This is because of the two separate signaling pathways, two linked signaling pathways in the chemotaxis system, which we're looking at to model it. The first signaling pathway is the faster set, and this is what controls the actual flagellar output of the system. The pathway is triggered by a complex that includes the methyl-accepting chemotaxis protein, KW and KA. KW and KA. The Ki protein has a chance to become autophosphorylated. And when there's no chemotactic attractor present, this happens with a certain rate. That phosphate group can then be transferred to Qy, creating phosphorylated Qy. Qy is broken down, is dephosphorylated by Kz, and this forward phosphorylation reaction and dephosphorylation reaction establishes an equilibrium in the cell of an equilibrium ratio of Qy to phosphorylated Qy. Qy and phosphorylated Qy then interact with the flagellar motor, and based on their ratio will change the run length. So in the normal system where you have the established equilibrium, which is about 2 to 1, you get run lengths of 1 second. When you add a chemoattractor, which we're calling a ligand in these models, it decreases the chance that Ki will autophosphorylate. This decreases the rate of phosphorylation of Qy and drives the system towards a new equilibrium point in which Qy is much higher than Qy phosphorylated. This triggers the flagellar motor to have longer runs and fewer tumbles. However, if this system was the only one that was present, in very high concentrations with no gradient, you would still get long runs. In order to prevent this and return the system to its normal movement, there's another pathway, which involves Qb instead of Qy. And Qb is phosphorylated in a very similar way based on the autophosphorylation of Ki, except instead of acting on a flagellar motor, it acts on the methylation state of the MCP. You may have been wondering why MCPs are methyl-accepting chemotaxis proteins. Well, the reason is that in the cytoplasmic region, they have 4 places where methyl groups can bind to. In the case of TSR, each methyl group increases the chance that Ki will autophosphorylate. And the methyl groups are removed by phosphorylated Qb and added by Kr. So when you have your equilibrium of unphosphorylated Qb to Qb, you're going to get an equilibrium state of methylation among your receptors. When you have the ligand binding, the phosphorylation of Qb decreases, and so your demethylation reactions decrease, and you get an equilibrium with a higher methylation state overall. Since the methylation state increases the chance that Ki will autophosphorylate, it counters the effect of the ligand and returns your system to the normal run length of 1 second. By combining these two reaction pathways, we can make a mathematical model that will let us predict the ratio of Qy to phosphorylated Qy based on the ligand concentration. Our model uses 17 differential equations and takes into account over 20 reactions and tracks all the different proteins that are involved in these 2 reactions. And the reactions are modeled using the law of mass action and Michaelis-Menten enzyme kinetics. And so here you can see at 5 seconds we introduce a step in ligand concentration, and the ratio of Qy to Qp responds by also going up, which is due to the fast Qy reactions. And then it's slowly brought back down to normal by the slower Qb reactions. And we're going to use this model to essentially characterize the ways that our chimeric receptors work. If we see any differences in the expression of chemotaxis, we can then use the model and effectively track it back to which parts of the chemotactic pathway have been altered. We'll use that to determine which of the different receptors that we produce we want to use in our final project. And we'll also use it to characterize the parts when we build them into BioBrick so that it's much easier for other teams to use.
All right, so an outline of our experimental strategy now that we've talked about the modeling of the system. First of all, we want to amplify the genetic components that we look to use in building our circuit, and create a ComP-TSR chimera library, and build a quorumtaxis circuit. And third, we want to screen for a functional quorumtaxis circuit. And so our first step is amplifying BioBricks. And if you look on top, this is the circuit that we hope to make. There's the standard cut sites, EcoR1 and PST1, a TEP promoter, and the ComP-TSR chimera in the BioBrick plasmid and transformed into E. coli. And so from our data, we have the correct plasmid and the TEP promoter isolated and amplified. Our second step was to create ComP and TSR BioBricks. And so we also have the COM parts isolated from B. subtilis and the TSR isolated from E. coli as well. Our third step is to create ComP-TSR chimeras. And there's two ways we could do this. The first is a rational design approach, where we look at the structure of TSR and the structure of ComP, and within the transmembrane domain, look for sites that are highly conserved and recombine at these sites. And with these chimeras, we can screen for function. On the other hand, we can produce these chimeras by way of a combinatorial library, where we take the TSR gene and the ComP gene, digest at various locations in the sequence, ligate these fragments, and then screen for functional chimeras with these fragments. Step 4 is to test for E. coli swarming. And the strain that we're going to use in order to screen for chimera function are these DEL MCPs. And this strain lacks the MCP receptors, even though it has the other chemotaxis components intact. And so our plan for the swarming assay was to spot the cells on a soft agar plate and assay for outward growth. And so for the wild type B. subtilis and E. coli, there is outward growth when they're spotted on these plates at 18 hours. You can see that the growth is blurry. On the other hand, the E. coli strain, DEL MCP, does not exhibit this outward growth even at a longer incubation time. And so our next step is to screen for functional chimeras. So with the DEL MCP E. coli that we just tested the swarming ability, we plan to transform these E. coli with the chimeric receptor and then plate these cells, or spot these cells on the soft agar plate with increasing gradient of B. subtilis culture extract, which would be the pheromone COMX that we hope that it binds to. And so if we are able to find a successful chimera, these E. coli will then swarm towards an increasing concentration of the COMX pheromone. So where are we in our work so far? Well, so far we have amplified the genetic components, the bio-breaks, and our new parts, the COM from Bacillus and the TSR from E. coli. So in the future, we hope to create the chimeras and build the circuit that was shown earlier and screen for a functional quorumtaxis circuit. And so far I've talked about our phase 1 of our work, which is to create the seek function. And the second part of our seek and destroy E. coli would be the development of a destroy output. And so far, we're looking at the COMX signal binding to the ComP receiver, and the output would be quorumtaxis. That would be our seek function. And then we hope to combine that with a different signal, a different receiver, integrate these two systems, and produce a signal or attack response through transcription. And this would probably be from B. subtilis SDPC toxin production. And so in finishing, I'd like to thank our large and diverse group. It was a collaboration of a lot of departments from Rice University and from Baylor College of Medicine. And thank you to our advisors for mentoring us and putting up with us. And collectively, we as a team are known as the Rice Taxis drivers. Taxis. All right, thank you very much. Questions? You guys want to come up? Watch out for the cord. Questions? Yes?
Why did you choose that particular toxin? And sorry if I missed it, but what are you trying to destroy?
Okay, so this particular toxin is produced in cases of nutritional deficiency. Bacillus produce this toxin when they're about to go into sporulation. So during nutritional deficiency, half of the population goes into an intermediate phase where they decide whether or not to sporulate. And so they produce this toxin in addition to immunity protein for themselves, killing the other half of the population that doesn't, and cannibalizes it. So we hope to use this toxin as a way to kill the bacillus. Does that answer your question?